Hello, everybody, and welcome to our third week of our COVID-19, the Nascent Impact course. We are excited to have you here, and we've had some fascinating talks, and today, in this week, we're going to continue that set of talks. Before we get started, I just want to make sure, first of all, that everybody had a wonderful weekend, and that you're watching the news, because one of the fascinating things about this class is that what's going on in the class is directly reflected in the outside world, and the kinds of things we're talking about week to week change even what the speakers are getting ready to talk about, because these questions are so immediate. So it's fascinating to be able to watch and to listen. A few updates about the class in particular. We've been grading your responses from last week, and they continue to be really interesting. There continue to sometimes be some challenges with citations, and I don't want that to take away from the content. So I wanted to remind you of a few particular things to strengthen your responses to the prompts. One is to remember to read the prompts before you see the talk and before you do the preparatory material, so that you have in mind the kinds of things you need to be thinking about. The other thing is the prompt is meant to, sometimes has multiple questions. The expectation is that you're cohesively, synthetically answering those questions. So it's not question 1, question 2, question 3 out of the prompt, nor is it only answer one of the questions in it, but you want to be using those questions as a way to be able to put together your response. If you are using materials, especially like in the last one, where if there's an expectation that you're supposed to go out and refer to an outside article and respond to it, please make sure that you are giving us the full reference. If you just say, I read a thing in the New York Times, we don't know what it is you read in the New York Times. You need to give us the full reference of the materials you're talking about. And that at this point is now an occasion to not get a satisfactory grade on a response. So you need to be very careful that you are, in fact, correctly citing. So those are some of the things, a reminder that if you need, in our Blackboard site, the FAQs have additional information about citation. You can always go there for reference, and they actually have links from there. And I want to make sure I talk about the citations every time, but I want to make sure that you're seeing that that's part of a cohesive whole. We want to hear, like, part of this is for you to practice your writing, to be able to think about how do you synthetically put together other people's ideas with your own and the experience of the world around you. So we really want to hear your voice as a part of that, but we want you to practice these other skills at the same time. One other thing I wanted to remind you of, and I added some information in the FAQ on the Blackboard site, the library is collecting, we're living through history as we're speaking right now, right? This is the kind of thing you're going to tell your kids and your students and your friends about, and you're all going to reminisce in 50 years to be like, remember that time? But you won't remember all the details. And Mason has been so affected that our library is keeping an archive of what the response was like. What was life like during the time of the pandemic and the shutdowns? And so there's a link that actually allows you to be able to update your own experience through the library portal to be a part of our permanent archive. It's kind of a StoryCorps through the library. And we would love for you to be able to add that. It's just an interesting way to be able to add your voice. So there's a link in the library for that. The last thing I'm going to remind you of is you've got the 2 buttons at the bottom. The chat is for you to communicate with the teaching assistants during the time that we're here about class details. The Q&A that you've got the button for is where you've got the chance to put your questions that you've got. And today we're talking about testing, right? This is at the top of everybody's mind right now. So the Q&A, I'm loving watching your questions coming through, watching y'all put them up and then voting them up, and then sort of keeping track of the kinds of themes that are coming through. But we want to make sure, I see one come up right now, we'll dismiss any questions in the Q&A that are about class, about the class structure. And that's the place we want to reserve for questions about the content. So, and you're welcome. And remember, as Tyler Cohen talked about, there's a speed premium. If you put a question in first, you're more likely to have it answered if you've got more people who are thumbs upping your question. So as you think about questions, go ahead and put them in, and then I ask them as we get towards the end of the presentation. So with all of that, I am really excited. So Ali Andalibi is the Senior Associate Dean in the College of Science. And he has a long history. He's a good friend of mine. And he's got a long history of combining research and public service and innovation and entrepreneurship. He's worked with NSF before. He's been working with overseeing how Mason is responding in terms of our testing for the the SARS-CoV-2 virus and our antibody responses, and he's overseen how the College of Science is responding. And so it's a real honor to have Dr. Andalibi here with us to be able to talk about this. So Dr. Andalibi, I hand it over to you.
Thank you, Dr. Usher. It's a pleasure to be with all of you today, and I hope that the next hour or so that we spent together, you guys are going to enjoy. What I'm going to provide you today is an overview of the common tests that are taking place for SARS-CoV-2. This is the virus that is responsible for the COVID-19 pandemic that we've been grappling with for the past few months. So with that, I'm sure that you guys know this, but as a reminder, coronaviruses, like all other viruses, are parasites, and they require a host in order to reproduce. Coronaviruses are relatively simple. They have in their core a single-stranded RNA that in this figure is seen as these beige squiggly lines. And this RNA is what encodes for the proteins of the virus at the end of the day. It has 4 main structural proteins, and this includes the spike protein that you guys, I'm sure, have heard, and that's the business end of the virus, or one of the business ends of the virus. The envelope glycoprotein, another membrane glycoprotein, and the nucleocapsid protein, which is shown here in the sort of the bluish color that interacts with the RNA. And all of these are encoded by the 3' end of the viral genome. There are several accessory proteins that are also encoded by this region. So the if you will, the genetic information of the virus is very, very compactly encoded. And this is in order to be able to save space for these very, very small parasites of human cells. As I said, bound to the DNA are the nucleocapsid proteins. And these are the ones that are involved in enabling the virus ultimately to replicate. What you see in the orange is the spike protein, and this is what gives it the crown-shaped appearance that the virus has, hence calling it the coronavirus. Sorry, I think I just, yes. The SARS-CoV-2 virus, like actually the other coronaviruses, basically enters the cell through attachment of the spike protein with a specific receptor on the surface of the cell, in this case, the ACE2 protein on the surface of the cells, and this is expressed in a variety of cell types in the body, including the lung pneumocytes. The virus then enters the cell. It releases its RNA genome, and a protein called the replicase is translated first. There are the non-structural proteins that are part of that complex, and what you end up getting is a replicase transcriptase complex, which then allows the synthesis of all the other steps to occur, and then you get the RNA synthesis follows next from the original material, and then there's translation of the various proteins that I talked about in the last slide, and the insertion of those into the endoplasmic reticulum. This is the ER Golgi intermediate compartment. And then you get the formation of the mature virions that are then released following exocytosis into as new virions, hundreds of thousands of them being released as they lyse the cell and begin to invade other cells. The picture that's to the right of the one with the in this picture gives you guys a little more of a detailed schematic of how the virus gets into the cell, replicates where it goes, and the other things, but you guys can study that yourselves. So now let's look at the virus. As you probably have heard, this COVID-19 has many facets. And the various facets of this disease are a direct result of the fact that the virus actually can infect and therefore affect a variety of cell types and organs in the body. So one of the ones that you guys have heard the most about is the impact of the virus on the lungs. What ends up happening is that as the virus binds to the epithelial cells in the lung tissue and begins to replicate, what you end up getting is severe damage to the tissues of the lung, the alveolus, the alveoli. So each alveolus begins to break down as a result of that. And what you get is a diminishment of the uptake of oxygen. As a result of that, the buildup of fluid that you see, the pneumonia that's associated with that, and the labored breathing that's often associated with the severe cases of the virus, and oftentimes, unfortunately, the result of death, especially in individuals that have pre-existing conditions. Sorry, that wasn't supposed to go. The virus also affects the heart and blood vessels. One of the things that we were seeing initially being a cause of death of this virus was the number of strokes that was occurring in individuals was rather high. People couldn't figure out why, but then this is because of the fact that the virus actually does bind to blood vessels and can impact the formation of clots, which are then, these clots are released and they result in strokes. There is definitely evidence of the COVID brain that you guys have heard about, and so there's good evidence that there is a direct impact on the central nervous system that could be leading to these kinds of conditions. You get conjunctivitis in patients, so this is very common that you see with a variety of respiratory viruses. Obviously, it binds to the nose, the nasopharynx, lung sort of tract is lined with epithelial cells, all of which have the receptors or the binding sites for this virus. And the virus can certainly bind to epithelial cells in the nose. And also, as I'm sure you know, there is impact on the ability to be able to smell things that's been associated and reported with this disease. And that probably has to do with the fact that in the nerve-rich areas of the nose where the virus binds, it also impacts the sensory cells that are in that area, and therefore prevents us to be able to smell things once we're infected with a virus. But this is, again, this is in a portion of the individuals. There is no such thing as 100% of symptoms being seen in everyone. Each one of these symptoms is seen in some groups of patients and not other groups of patients. Another area that's affected, or another organ that's affected, is the liver. And we oftentimes see an elevation of liver enzymes in patients with COVID-19. And that could be associated with not only the impact of the virus directly, but also because of all the meds that are given to patients who are suffering from COVID-19, and usually medications, medications are broken down by the liver, which is the major detoxifying organ of the body. The kidneys are oftentimes damaged by the virus, and this is one of the another one of the important reasons why people become very, very ill as a result of the virus. And so taking care of the kidneys is an important clinical, if you will, part of the clinical management of the disease. There's a lot of reports of diarrhea, and although it's not present in all of the patients, it is certainly present in a fraction of the patients. And we do know that the virus is capable of infecting the epithelial cells of the intestines, and the virus can therefore be passed in feces and has been detected in feces of individuals. So that could be another route of transmission for this virus that has not only tropism for a variety of cell types, but also has, as a result of that, the ability to be transmitted in a variety of ways. Although the most important manner of transmission is through coughing and the production of droplets, and more importantly, in my view, of aerosols. So as I mentioned in the last slide, the types of symptoms that have been observed in this disease do not occur in all the patients. Fatigue is something that is seen in about 80% of the patients, that's followed by loss of ability to breathe. Joint pain is seen, vision problems, cough is an important one. That's over 60% of the people have it. As you can see, around 50% of the people do lose the sense of smell. There's a loss of taste as well, and headaches and lack of appetite, muscle aches. These are, the headaches and lack of appetite and muscle aches are very similar amongst respiratory viruses. And then there is the fever that's associated with it, with the virus, that's oftentimes one of the most important and also common symptoms of the disease. And as you can see, the fatigue and the shortness of breath is something that actually stays with people, although hopefully with new treatments and new treatment regimens, we'll be able to prevent the presence of these symptoms in patients as once they've been cleared of the virus. So one of the things I want you guys to consider is that the death rate, as well as the health conditions that are associated with the death rate. The death rate of this virus is relatively high in people who are in ages 60 and above. Particularly high in people who are older, so grandparents, or even elderly parents and so on, are very, very potentially susceptible to disease, and therefore making sure that these individuals are as safe as they can be with making sure that you as young people practice all the safety and precautionary measures that would help you not contract the disease and therefore not pass it on to your beloved elderly family members. As you can see, cardiovascular disease, diabetes, pre-existing respiratory disease, hypertension are very important contributors to the mortality of this disease, and we've seen that in studies from around the world. So comorbidities are definitely an unfortunate part of of making a person really, really sick from this disease. And this is probably one of the reasons why younger individuals who have not yet developed the comorbidities are more or less likely to succumb to this disease than older individuals who may have developed it as a result of just simply aging. So now let's go to, so I gave you all of that to put into context why we and how we test for this virus. Obviously, the why is in order to identify patients who are actively infectious, but also to identify patients who may have been exposed in the past, say young people who may not have had severe symptoms or who may not have had any symptoms at all. But we need to know that because that could be an indicator of whether or not the individual actually has protective antibodies against them, against the virus if they have been exposed to it in the past. Looking at the tests, the assays that I'll be discussing today, RT-PCR is the gold standard that's being used today. It's a laboratory-based assay, and it's very highly sensitive, and it's used in the US and elsewhere quite extensively. LAMP is another technology that is also not as commonly used, but I'll go into it in a little further detail. It's like RT-PCR is a DNA-based technology, an amplification-based technology, and is also very highly sensitive. The lateral flow tests, there are a number of them that are currently on the market, and these are designed to be a very, very rapid and relatively inexpensive tests for determining the presence of IgM and IgG to the virus in an individual. So they're more of a screening test and a test for determining what percentage of the population has been exposed to the virus. Similar protein-based technology is the ELISA assay, and the ELISA assay can be used to measure antigens or antibodies. But in this case, we're going to focus on the antibody-based ELISAs, or ELISAs that are looking at the production of antibodies in individuals against the virus. So let's dive into the first one, which is the RT-PCR assay. And that is the most commonly used assay to determine whether or not someone has COVID-19. So let's say someone is not feeling well, is experiencing a fever, malaise, and just overall something that would, a state of health that would make them wonder whether or not they've been exposed to the virus. They go to their doctor, or they call their doctor, and the doctor says that you need to get tested. And RT-PCR is the test that's done. It is truly the frontline test, and the test, the RT-PCR test detects are regions of the virus's genetic material. The spike protein is common, the nucleocapsid, the envelope protein are also used to detect whether or not the virus is present. This is a very highly sensitive test, as I mentioned, because the primers that are used for the assay are very, very highly selected and selective primers for COVID-19. So basically, once you collect the samples either from the nose or the back of the throat, so basically the nasopharynx, you could get the samples from other anything else that would, where you can get live viral particles from. But once the sample has been collected, you get rid of the proteins and fats and all the other molecules. You're left with the RNA. That RNA is copied into a DNA, and then you amplify that DNA using PCR. Nowadays, fluorescent dyes are used, and RT-PCR, as the technique is commonly called, uses the production of the fluorescence or an increase in the fluorescence of a sample as a way of determining whether or not a sample is positive, and also as a way of determining what the original copy number was when in the sample that was taken from the patient. So here's a schematic of the process. You collect the sample from the nose or the back of the throat. You extract the RNA. You reverse transcribe it. You make the DNA, and then this DNA is amplified using specific primers. In this case, fluorescent probes are used, and the DNA polymerase comes, and as it goes down as it's making the new strand, it displaces the dye, which has a quencher. You guys don't need to worry about that, but as the dye is released, its fluorescence increases, or it's allowed to fluoresce, and as more and more copies are made, as you can see it here, the fluorescence begins to build up, and once it passes a certain threshold, you consider the patient to be positive. So what does the result mean? Well, a positive test result means that the patient is currently infected by the virus. But a negative test result always leaves you scratching your head, because one thing, it could be that the patient is actually negative. I mean, so the person is not currently infected by the virus, or that wherever you took the sample from did not have any virus on it, or that the sample that you took was, was a poor quality sample. The sample was left in the car at, on a hot day or something like that. The sample just isn't good, and the material broke down. So you have to confirm the negative result to ensure that the patient is indeed negative, and that's why you do it a few days later. You do a follow-up test with a few days later. What this test does not detect is if a person has had the virus and then has been cleared of it. It really is meant to only test the presence of the disease. So if you had, if you had a really bad, flu-like set of symptoms, and 3 weeks later, after you've recovered, you decide to go and get tested by RT-PCR for COVID-19, you can't say that you had COVID-19. The answer would not be able to, this is not the test to give you that response or that answer. So the other test that's used, this is actually a relatively simple test. It's called loop-mediated isothermal amplification, and LAMP is around for a long time, and people have used it in a variety of settings. But being a test that's relatively easy to do in settings where you don't have access to PCR machines, but you do have access to primers. Some people have been using LAMP, and it's something that I wanted you guys to be aware of. The PCR process requires the cycling of between temperatures that are high and temperatures that are relatively lower. So what you do at the high temperatures, you denature the DNA. Then at lower temperatures, you anneal the primers and you extend those primers to get the amplification. With LAMP, it's a little bit different. You use the same temperature, and what you use are 6 sets of primers and what's called a strand displacing polymerase that once the primers anneal, and they begin to extend. As the polymerase extends it, it displaces the other strand, and you can anneal the primers on the other end, and you next can use what are, I'll show you in the picture, but through this cycle of adding the primers going to one end and then the other end and forming loops, you can actually get very, very high levels of amplification of the viral DNA. With 35 cycles of PCR, you get somewhere in the neighborhood of 33 billion, 32 point some billion copies of the DNA of the original material. In the case of LAMP, that number is actually higher. So you can actually see it with the naked eye because the number of copies is so high. And so some people use magnesium pyrophosphate as the end product. Other people use a dye. So there are a number of ways of doing this, but I'll show you the design of the primers. I'll show you schematically. So you have forward primers, the F1, F2, and F3, which are for the 5' end. And then the backwards primers, which are B1, B2, and B3, which are targeted towards the 3' end of the target region. So you denature the target DNA, and then you have this F1 complement primer, which binds to the F1 region, plus the F2 that's attached to it, and you get, you bind this to its target, and you get extension, and you get this double-stranded DNA. Then you put the outer primer in, and you get the reverse of that happening. And this goes on, and then you get the loops that where F1 and its complement bind, and B1 and its complement bind, and each one of these forms this dumbbell shape, which is what really, if you look at it in the next picture, it'll show you how this amplification is done. So as you can see, here's the F3, F2, and F1 primers. Here's the example of the F2 with the F1 complement, and the B2 binding to its region with the B1 complement, and the amplification that can occur as a result of this formation of this loop and this loop. And then you have loop primers that actually increase the number of amplifications that can occur. And hence, there's a great YouTube video that you guys can watch that shows you the how this amplification, LAMP amplification is done. So the positive result, very similar to PCR, means that the person is currently infected. Negative result could mean that the patient is not infected, or that the virus was not present. It's too late, too early, the sample was bad, very similar to what you see with PCR. So now on to our lateral flow test, which is the test that is commonly now being used for looking at patients and their exposure in the past to the virus, because the antibody tests are looking at the presence of a response to the virus, an immune response to the virus in the form of initially IgM and later IgG. So LFAs, lateral flow assays, these have been commonly used, called antibody tests. You can use it for antigens, but everyone, all the tests that are currently available are antibody based. And what they do, as I said, is test for antibodies in the patient's blood, saliva, or any other fluid that you want to get the sample from. And so what you put there's a, I'll show you in I think in 2 slides, but you take some of that material, you add some diluting buffer to it, and the material begins to flow, and it binds to the appropriate sites on the assay kit or the assay platform, and you see the formation of bands. This is the same technology that's used for pregnancy tests. So but there are a lot of YouTube videos also available on this. Here it is. So here's the sample pad. You put your blood or saliva on there, and you add the buffer, and things begin to flow. And at this point, you have this is a colloidal gold. So it's just, these are gold particles to which the antigen, in this case, the viral antigen, has been conjugated. So as the antibodies against COVID bind to the COVID antigen, they go and, depending on the presence of anti-IgM antibodies or anti-IgG antibodies, those specific antibodies bind to their regions. And if there's nothing, if there's no antibody, no IgM or no IgG, you have a positive control that goes and binds to the positive control line that will tell you whether or not the assay was working. But basically, if you get an IgM positive, you get the first line and the control line. If you have an IgG positive, you get the second line as well as the control line. And if you have both IgM and IgG present, then you get both lines lighting up the IgM and IgG line as well as your control line. So again, here's a link to a YouTube video where you guys can take a look at this. Here's a typical what an assay or a device would look like, again, with the sample well and the presence of the IgM, IgG, and the control lines. So what does the result mean? So that means that if you are positive, if you're IgM positive, means that you were recently exposed to the virus. If you don't have IgG and have IgM, that means that you've been recently exposed to the virus. Although I have to tell you that with COVID-19, the timing of IgM and IgG is not the traditional timing or the typical timing that we see with other things. And I have seen cases where there's a co-development of the IgM and IgG response in patients. So, but typically it's, if you see IgM and not IgG, that means that you were exposed to the virus relatively recently. At this, in that case, you can actually, you may be infectious, and if they were to do an RT-PCR test on you, they would be able to see that you are positive. If you have IgG and no IgM, that usually indicates that you were exposed to the virus some time ago, and that you have mounted an immune response, an IgG response, to the virus, and most likely this is what protects you from the virus in the future. This is what you see when someone has been exposed to any disease or has been vaccinated. You have an IgG response. The IgG level of antibodies is high, and you'll be able to detect it if you were to run an assay such as a lateral flow assay. This this gives you an so that's the idea behind the lateral flow assay. And enzyme-linked immunosorbent assay, the ELISA assay, is the assay is very similar conceptually to a lateral flow assay from the molecular side of the if you look at the molecular aspects of the assay, i.e., you have antibodies against a particular antigen, and you want to detect the presence of those antibodies. Now, in this case, using an enzyme-linked immunosorbent assay. Why is it called enzyme-linked? Because you have enzymes that are attached to antibodies that can attach to your molecule of interest, and then those enzymes can cause the production of or catalyze the production of some sort of a indicator. Either color or in some cases light or so on, that can give you an idea whether or not the test the sample is positive or not. So you have several forms of this. One is called the direct assay, where you take a well, you coat it with a particular antigen, and you have an antibody that has been derivatized with an enzyme, and then you add the substrate, and if the antibody has bound, and then of course you wash the wells, and if the antibody has bound to the wells and you add the substrate, the enzyme acts on the substrate and you get the formation of let's say, the color. In another one, the indirect assay, you have an antibody that, let's say in this case, would be the antibody against COVID-19. So this would be the patient sample. You come in and it binds now to the antigen that you use, let's say the spike protein antigen. So now these antibodies are bound. Now you come in with a secondary antibody. So let's say it's a goat anti-human IgG antibody, and that is linked to an enzyme, and then you add the substrate, you get the formation of the color. And then the third one is a sandwich assay, where you have a capture antibody that has that, let's say that you make antibodies in bulk to, let's say the antigen on the surface of the virus, let's say the COVID-19 spike antigen, and then similarly you look for the presence of the antibody to that antigen, and then you come in with an let's say a goat anti-human IgG antibody that's been derivatized with an enzyme, and then you add the substrate and you get to see the color. The results very similar to the LFA. Means that the patient has antibodies, and if you don't have antibodies, you haven't been exposed to the virus, i.e. you have not been infected. So this just gives you a schematic of what is used. So initially, let's say that this is the start of symptoms on day 0. You have a window where you can do your nucleic acid tests. As over time, the viral, let's say in the nose, the virus begins to drop, but during this time, what you begin to see is a rise in the antibodies, and later on during the illness, you can actually test for antibodies. Here's a more more busy but a little more informative picture of this that shows you the nasopharyngeal swab PCR, the isolation of the virus from the respiratory tract. This is direct isolation and using plaque assays to see whether or not there's virus there. Bronchoalveolar lavage can be performed, and as you can see, that that goes down more slowly over time. Stool PCR, as I said, you can often see the virus in the stool, and then the presence of the IgM and the IgG. And as you can see, actually, these do go up relatively simultaneously. So there is a difference between this virus and some of the other viruses. Now, I think then here is a summary slide for you guys to look at this on your own, that compares the LFA, the lateral flow assays, these rapid serology tests with ELISA, which is a lab-based assay, versus the diagnostic PCR. The LAMP assay would be similar, and what the inputs are, what the result outputs are for this, and what the strengths are, and what some of the limitations are of this, or of each one of these assays. As we do more testing, as we learn more about this disease, we're beginning to learn about the pros and cons of each one of these assays, and we're still learning about this virus, and I'm sure that we'll be learning a lot more using the assays that are available to us today, and assays that will perhaps be developed in the future to track the virus. With that, thank you very much, and now I will answer any questions.